I'm working on reconnecting to the VPN. My computer fell asleep. Okay. If you want to prevent that, if you open up your system preferences, and if you go to Energy Saver, you could tell it for your battery to turn display off. Obviously, I have mine set to never to avoid my computer sleeping and my processes stopping on me. So you could do that for yours if you want to avoid that in the future. I change it for both my battery and my power adapter because I'm usually plugged in. So you could just put that all the way to the right or to 3 hours or something like that. And it should prevent your computer from falling asleep. So as Christina gets her VPN back up and running, the data we're going to be analyzing, can you explain why, how Trimglor was picked over, say, Trim-O-Matic, please? Good question. Honestly, there's no real rhyme or reason to it. One reason that we picked Trimglor, it uses CutAdapt under the hood, but one of the convenient things about Trimglor is it's a wrapper, so you can actually run trimming and then subsequently FastQC all with one command. So that was really the main reasoning behind picking it. But in fact, we actually don't do that anymore because the naming that's generated from the trim files for paired end reads says like Val or something weird in it, which isn't horribly informative. So after selecting it with that purpose, realizing that it would make more sense to change the name of those trim files to be more informative. So we actually don't use that functionality of it. But initially, that was the reason why we selected Trimglor. If you want to go with Trim-O-Matic, it pretty much does the same thing. I think it's just written in a different coding language. And again, this luckily wasn't one of the steps. So some of the steps in this pipeline were like highly debated among the analysis working group members. But some steps weren't horribly debated. So this one, our analysis working group members were basically fine with anything, just because they all pretty much do the same thing. But some of the other steps, we've had heated debates. Like it was literally about 1.5 years of just talking back and forth and setting up meetings with the scientific community through our analysis working group members before we settled on a pipeline that everyone was happy with. So people tend to like to go with what they know, which it makes it difficult for people to be flexible about changing things with the most current, most up-to-date or best tools. But the pipeline we put together is actually pretty robust. I like it a lot. So when I first started, remember, I'm all self-taught. So when I was first started, I knew very, very little. So I really relied on those analysis working group members to help put the pipeline together. And since learning more and more about all of the steps, we actually put together a pretty good pipeline. But yeah, I actually work on a COVID-19 project as well. And for that, we use Trim-O-Matic. Okay, Christina, you're good to go. Is everyone else good to go with their Jupyter notebooks? So this Jupyter notebook, again, was built as a San Jose State collaboration with us at GeneLab to teach all of you how to process RNA sequencing data using our GeneLab standard pipeline. So in this Jupyter notebook, you're going to see step-by-step instructions for processing samples derived from the soleus, or also known as the anti-gravity muscle. So this is one of the small muscles that make up the quadriceps. So the quadriceps are a group of muscles, your calf muscles. And so part of that group of muscles is the soleus muscle, and that muscle tends to change the most in the context of unloading, so in the context of microgravity. And so we're going to be looking specifically at the soleus muscle of mice that were either flown in space aboard the International Space Station or a respective control group that was kept on Earth in an environmental simulator to actually mimic the environment of space, so mimic the humidity, the carbon dioxide, the light cycles, stuff like that. So the goal is that the only difference between our spaceflight and our ground control group is the actual space environment. So microgravity, exposure to radiation, stuff like that. So we control for everything else. And that was during the Rodent Research 1 mission. So the very first mission that we housed mice on the International Space Station before my time. That took place in 2014. And then if you guys want to read more about the samples are more about the study, here's the link to our GeneLab repository, and then specifically here's a link to the data set, GLDS 104. And I kind of showed this to you guys before during the GeneLab overview lecture, but this is specifically GLDS 104. So if you guys want to learn more about more than what I just told you, you can go ahead and read on the study description. And then there's also been a few publications. There's also been a few publications from this mission. Okay. I am going to turn off my camera now. We can start my report, yeah. Okay, so what this notebook covers is it's going to start off with raw reads, so those FASTQ files that we took a look at in the lecture, and then we're going to assess the quality of our raw reads, and then we are going to trim and filter those raw reads, and then we're going to take a look at the quality of those trim data. So that's probably as far as we'll get before we hop to lunch or before I transition to a lecture. And then we are going to map those reads back to a reference genome to find out where they came from. And in doing that, we have to create an index. I'll talk about that during the lecture. And then once we map those reads back, we're again gonna use a program called MultiQC to take a look at the alignment data, so to see how well we mapped back. Oh, and Marie just put in a link to the Rodent Research 1 mission if you guys want to learn more about that specific mission. Thank you, Marie. And then after we map the reads, we're going to count them using a tool called RSEM. And then finally, we are going to put all of the counting information for all of our genes, for all samples together in one nice big table to take us into tomorrow. So hopefully we'll get through all of this today. But again, if not, we do have additional buffer time. So it's okay if we don't get all the way through this notebook today. Okay, let's get started. So just like our intro to Unix, well, I guess before we get started, does everyone see the Python 3 idle down here? Is there anyone who doesn't, I should say? Okay, I'll take your silence when you guys are all seeing Python 3 idle. So this is using the Python kernel from the Jupyter notebook. But similar to our Unix intro that we did, the commands that we're going to be running through here are going to be all Unix commands. So you'll notice we'll be adding those exclamation points at the beginning, except for the final part where we generate the raw counts table. That we'll do in Python. So I know a lot of you guys have Python background, so that code should look familiar. So if you want to navigate to any part of the Jupyter notebook, you can of course click anywhere here on the table of contents to bring you directly to it. But we're going to go through each step at a time. And again, if anyone gets too far behind or needs some extra help or drops off and has to come back, we have our assistants on deck to take you guys into a breakout room to catch anyone up who might miss some stuff. But I'm hoping, just like with our other notebooks, we'll be able to keep most of you guys, if not all of you guys, together because we did account for buffer time if people have questions or issues. Okay. So the first thing we want to do, I'm an organizational freak. I tell people that being well organized is a gift to your future self. And I take that philosophy in all aspects of my job, or at least try to when time permits. And so I think it's very important to talk about how to set up the data we're going to be generating. So we are going to be generating a lot of data. In a lot of data files. And so the first thing I like to do is set up a directory structure to keep all of the files that we generate very organized so that we know where to find stuff, where we're going to output stuff, and whatnot. So you guys are going to be using that make directory command to create the directory structure that we use at GeneLab when processing RNA sequencing data. So the first thing we're going to do is run this first code block here, and we're going to make a directory in the title. We're going to make a directory in our current directory. So that's that dot shortcut you guys are used to seeing. And we are going to name that directory GLDS 104, which is of course the name of the data set. So you guys can all go ahead and run that code block. And when you do, you should actually see your GLDS 104 folder being generated on the left there. I'm gonna go through. If you guys ever get behind, just please message the chat and I'll stop and slow down. Okay, so these next series of commands, now that we've created our primary directory, GLDS 104, we're gonna create subdirectories within GLDS 104. So this next code block is gonna create subdirectories for all of our raw data. So we're gonna have a subdirectory with the raw data title, and then we're gonna have a subdirectory within there to hold our FastQC reports that we're gonna generate. And then I didn't talk about this in the lecture, but there's a nice tool that allows you to compile log information or quality reports, and that tool is called MultiQC. So we're going to use that tool to kind of combine all of our FastQC reports to look at them all together, and we're going to put that information in another subdirectory. So if you guys go ahead and run that code block to make your raw data directory structure. And then I add numbers here just so that we know which step it is. So 00 is our raw data step. That's our initial step. And then we're going to go, you'll notice that each step increases the number, just so that way I know what order the files should be generated are. So in our next code block, we are going to make all the subdirectories for our trim data. So go ahead and run that. And I name the folders as descriptive as possible without getting too crazy long. So you'll notice that TG stands for trim galore to remind me that I'm running trim galore when I do my trimming. And then preproc stands for pre-processing. If you guys remember, I mentioned that when you do your trimming and filtering, it's known as pre-processing your data. And then you'll see the same set. We'll generate new FastQ files that are trimmed files and then do our quality controls. Next, we are going to create our directory for our alignment or our mapping. And so we use a tool called star. That's why you see star_alignment here. So go ahead and run that code block. And then we have another subdirectory of where we're going to store all of our count data. And we use a tool called rsem to do our quantitation. So you guys can go ahead and run that. And then as kind of we were discussing with the point that Felix made with accounting for different transcript lengths and whatnot when analyzing our data, we will, after we do our counting, we're going to normalize that count data. And so we create a subdirectory for the normalized count data. So you guys go ahead and run that. And then lastly and most excitingly, we are going to create a subdirectory for our differential gene expression. And so you'll notice that the normalization step as well as the differential gene expression happens with the same tool called DSeq2. So you guys go ahead and play that. Now, as I mentioned, RNA sequencing data is very large. The data products that are generated are very large. Some of the processes can take a while to do. And so to avoid all of us having to sit here and wait for everyone's job to finish, we would be here for weeks. And so to avoid that, I processed all of this data ahead of time. And so anytime that we run a job, we're going to be pulling our input data from the set of process data that I already generated. And so in order to know where to pull that data from, we are going to create variables for all of the levels of data. So similar to what we did in our Unix command where we created variables for paths, we're going to be creating variables to the paths of where all of the data that I generated is stored on the San Jose State cluster. So go ahead and run that code block. Okay, now that we got all of
do we have to change our username in all of the lines? Sorry. Because it says AS Butler.
No. So I generated all this data. So where I generated the data is in my scratch space on the cluster. And so that's where you're going to be pulling the data from. So you want to run this exactly as is. Good question.
Thank you.
Yeah, so all the data that you guys are going to be generating during the course are going to be in your home directory. So that's kind of the directory structure we just set up here. So all the data that we're actually going to be processing during the course is going to be going into your home directory. But then kind of this primary set of process data that I have to avoid us having to wait for all of your jobs to finish is going to be here. And so we're just going to pull the data from there. So yeah, you want to keep this as is. Good question. Okay, now that we got all of our housekeeping out of the way, we got a nice directory structure all organized and ready to have output data put into it, let's get started. Okay, so our first step, of course, is our raw data quality control. And so first, we're going to assess the quality of the raw sequence data from GLDS 104 using the FastQC program. And as we just discussed, short-read sequence data are stored in FastQ files. So I just have repeated here that 4-line structure. And again, reminding you guys of what each line indicates. So since we just went over that, does anyone need a refresher on that? Are you guys good with the structure of FastQ files, that 4-line structure? I'll take your silence to mean you're good. Okay, so what we're going to be doing is using this FastQC program to evaluate the information provided for each read in the FastQ files and then generate a summary report about the quality of the sequence data. So this is just like what we just took a look at in the lecture material. And of course, assessing the raw sequence quality is going to be important for determining if and how those reads need to be trimmed. So as we're going through this course as well, most of these things, again, because we don't want to take up all the computational resources on the cluster, we're going to be processing one sample in real time. But then again, we're going to be pulling the data from all of our samples. And so I probably should have pointed out here, if we go to the sample table, we can see, take a look at our columns. I'm just going to do source name here, sample name, and then our factor values, space flight. Okay, and so as you can see here, these are the data that we're working with. So these are all the samples. So we have 6 ground control samples that we're going to indicate as ground control, abbreviated GC, and then each replicate, that's a soleus muscle that came from a different mouse. So these are considered biological replicates. And so we're going to have 6 biological replicates from ground control animals, so animals that were kept on the ground. And then we're going to have 6 biological replicates from spaceflight animals. So those are the animals that were flown on the International Space Station. And we abbreviate spaceflight with FLT. So what we're going to do for the FastQC analysis is we are going to run FastQC on the forward read of one of our flight samples using the following command. However, you guys are not going to run this command directly because remember, we're all logged into the head node right now. And if you guys remember from my introduction to command line lecture, San Jose State is set up as a cluster, so anytime we want to run a job that requires a lot of compute resources, we don't want to run that on the head node. So we're going to want to run that on the compute nodes. And in order to do that, we have to submit our job to that job scheduler that'll make sure that there's enough resources on those compute nodes and allow our jobs to start and to run. And so because FastQC can take a little while to run, it doesn't require a crazy amount of compute resources, but since there's so many of us running it at the same time, we're going to be submitting it to the San Jose State job scheduler called Slurm. But this is what the command we are running looks like. So what we are going to be running is we are going to be running FastQC. We're going to be using this -o parameter or flag. And that is going to indicate the output directory, so where we want those FastQC results to store. And we're going to put that in that folder that we made up above, so that FastQC subdirectory of our raw data directory folder. And then the last argument in here is a required argument, and so this one is our FastQ file. And you'll notice that the FastQ ends in .gz, so these are all compressed using a gzip program that's built into Unix. And so all of these raw data files are compressed because they're huge. And so a lot of the tools that we use will be able to take in compressed files. So within the tool itself, the first thing that it does is it uncompresses the FastQ file. And so we want to tell our FastQ command where that FastQ file is. And so this is As I said, all of your input data is going to be pulled from that set of data that I already generated. So those are all of the variables that we just created in here. So as you can see, the raw FASTQ variable is where the raw FASTQ files are held for that data set. And so that's what the command looks like. So the input data for this command is going to be our raw FASTQ files. And then the output data is going to be what's generated from the FastQC program. And we're going to get an HTML report, and that's what we just went over in the lecture. And then we're also going to get this zipped directory. And then if you uncompress that directory, what you'll see is all of the files containing the information that's used to create the nice graphs that you see in the HTML report. And so for the Slurm job scheduler, to submit that command, what I did is I made a script called raw fastqc flight just rep 1, because remember we're just doing one sample, and I ended it in .slurm, so that way I know that that script is written to be submitted to the Slurm job scheduler. And so your $ processing, that is going to hold the variable here, and that is where I put all of the scripts that I made that we're going to have to submit through the Slurm job scheduler. And basically what that script has is, in addition to some commands that are important for Slurm, it also has this command in it. So this is the command that it's going to be running. So the command that it's going to be running is the one that we just went over. So that's what's inside that script there. And then in order to submit stuff to the Slurm job scheduler, we run a command called sbatch. So sbatch tells the San Jose State cluster that that I'm giving you a script to submit to the Slurm job scheduler. So you guys can all go ahead and press play. And you should see, could we cat the Slurm script to see what it looks like, please? Of course we can. So if you guys ran it, you should see that your job was submitted, and it'll give you a job ID number. And we can go ahead and add a code block underneath it. So again, I just use that plus sign to add a code block underneath it and to take a look at what that Slurm script looks like. Don't be too scared. I add a bunch of stuff to my scripts to do some housekeeping. So we can go ahead and cat that Slurm script and press play and run it. And so this is what the actual script looks like. So up here, bin bash. So if you guys can recall, our interpreter that we're running on our terminal is bash. So we want to let Slurm know what our interpreter is, so that way it knows what language these codes are written in. So that's the first thing that it is. And then everything that starts with hashtag sbatch is specific for the Slurm job scheduler. So I give the Slurm job scheduler a job name. I tell it to print all of its standard out to a file titled with the job name .out. I tell the Slurm job scheduler how much memory it needs. So this allows it, right? Because we have to tell it how much memory it needs, because then the Slurm scheduler is going to look at all those compute nodes, and it's going to see which one of those compute nodes has that amount of memory available. And then there are different partitions. So as you're running your Slurm job scheduler, you can, so thank you, Jonathan. The memory is in megabytes by default. Good point, thank you. So this is 8 gigabytes is what I reserved. It's actually more than what it needs, but I always like to be on the safe side and reserve more memory, because if you don't reserve enough memory, it will crap out. It will die. How do you know how much memory this file needed? I Google it. So I Googled it first to see how much memory this particular program takes based on the amount of input data. And then I try it out. So whatever I find during doing research on the internet for how much resources it needs, I usually add a little bit just in case. And then I run it like a test for just one sample through my system. And then if it works, great. And then I'll apply that to all of them. If it doesn't work, then I'll increase it if it ended up not having enough resources. Great question. And then there are tools and stuff, and actually within the Slurm job scheduler, you can set it up so that way it will tell you exactly how many resources your job used. And then you could use that information, excuse me, to fine tune how much resources you request. So there's a whole bunch of ways to optimize to make sure you're going with the right amount of resources. And why you want to do that is because getting into these partitions, so the Slurm job scheduler is set up to prioritize jobs in a certain way. So basically, if we have a bunch of people submitting jobs, and if someone submits a job that needs like 500 gigabytes of RAM, awesome. So Phil just checked the HPC and everyone's job is running. Wonderful. And so how the job scheduler works is if someone needs like 500 gigabytes of RAM and they submitted their job ahead of you, but your job only needs like 50 gigabytes, then what happens is as soon as 50 gigabytes are available, that 50 gigabyte job, even though it was submitted second, it'll start running. So that way it's not wasting compute resources, and compute resources aren't just sitting there. And you can set up, so this is stuff that Steve Boring has done for you guys, setting up the cluster. You can also set up certain partitions. So let's say you have a really high priority experiment that you're doing and you like need your data right away. You can kind of talk to your system admin and see if they'll let you, see if they'll set up a partition so that way your job goes to the head of the queue. So I actually got the privilege of doing this working on COVID work because, of course, obviously in the middle of a pandemic, we really wanted to get those data as soon as possible. And so we had a partition created to be able to jump to the head of the queue. So the partition nodes on the San Jose State cluster is just the normal partition that everyone submits their jobs to. So those are all you'll notice that for all of the Slurm scripts that we run, it'll have the set of sbatch commands. And that's what it is. It's talking to the Slurm job scheduler and telling it what it needs. I always add this stuff to my script. So if you guys remember the date command that we ran when we were doing the Mike's tutorial in the binder. So the date, I have it printed in seconds, and I store it in a variable called start. And then I echo, so I return what the start is, which is again is the date in seconds. And then I also want to know what node in the cluster my job's running on. And so that's stored in a variable called hostname. And that variable is a variable that's internal to Unix. And so this will allow me, because in a cluster, you know, in the picture of the cluster that I showed you guys in the intro lecture, I had 3 compute nodes called Snap, Crackle, Pop. I don't think those are the names of the nodes on the San Jose State cluster. You can name your nodes whatever you want. And so what this will return is it'll tell me which of the compute nodes that it was run on. And the reason that I do that is so that if, let's say, I run a bunch of jobs and I notice that, let's say I run like 20 jobs and I see that 5 of my jobs fail, I can go back and look at my output and see which node those jobs were run on. So if all 5 of my jobs that failed were run on the same node and all the ones that were run on a different node finished just fine, I might talk to the system administrator and say, hey, is there anything going on that's wrong with this node because all of my jobs failed that were run on it? So that's why I report what the host name is. We then need to activate the region that contains the tool that we want to use. So I went ahead and installed all of the same tools that Stephen installed that were being used on the Jupyter notebooks. So I installed those same tools in my local area on the San Jose State cluster. So this is where all the tools that I use to run RNA-seq are located. So I need to activate that location so that way the computer can find the tools, or the cluster can find the tools that I'm trying to run. And then I tell the computer where to find the input data. So I store a variable called in-directory for my input data. And that's that location that you guys all just made a variable for too. So that's where all my input data is. And then I create a variable for where I want my output data to go. So this is saying print the output data in my current directory in GLDS 104.00 raw data fastqc reports. So that's the directory that all of you guys just made. So that's why your output is going to be printed in your home directory there. I then run commands to tell myself what job I am running. So I echo the fastqc, and then I also want to know what version of the tool it is. And so all tools, they're continuously updated, right? So tools that are maintained, the authors of those tools will continuously update those tools to keep up to date with the latest technology and also the latest advances in bioinformatics. And so if you run the same set of data using a different version of the same tool, you could get different results depending on what the differences are between those tool versions. So I always like to keep track of which tool version I run. And so I make sure that I spit that out in my standard output. So that's what these commands are. And so similar, remember how we ran tools and we would look at like the help menu, -- help. Also, a lot of the tools have a command that's -- version. And so that way you could spit out the version of the tool. And then I just put some spaces in here. So echo, those blank quotes are just to kind of space things out. So this is not all jumbled together. And then so this is the command that we are running. So you'll notice that command is exactly this, right? So that's the actual command that's being run on the data. And I actually store that in a variable as well. So I store that in a variable called call. And the reason that I do that is, so I store it like as a string in a variable called call. And the reason that I do that is so that way I can echo or print out the exact command that I ran. And so in the standard output, when you do this, it'll print out the exact command that's run. So that helps with troubleshooting. So then when I go to troubleshoot, I could make sure that was indeed the input directory, the directory that I set the variable for, was the output the directory I set the variable for, did this run what I wanted it to run? And so that's why I do that. And then Unix has a built-in program called Eval, and that basically just says, hey, that's actually a command, please run it. And so this part is running the command that's held within that call variable. And then, so once the command is done running, I'll give it another space, and then I always end with this set of commands. And so this basically, I print out the date again, but I print out the date in seconds, and not this time it's at the end of the run. And so I then, so I store that as a variable called end, and then I print out that end. So I know the end time, which is the date in seconds. And that allows me to calculate how long the job took me to run. And so then I calculate the runtime in seconds, because remember my start date is in seconds, my end date is in seconds. So then I do my end date minus my start date to give me my total runtime in seconds, which I then print out. And then I have a variable to say the number of seconds per minute and the number of seconds per hour. And then these are just doing the calculations to calculate how long this job took in minutes and in hours. So I end up printing out the time that the job took in seconds, minutes, and hours. So I know that was a lot of information. You can blame Felix for asking to see the Slurm script. And if you guys have more questions on that, we could talk about it during the break or offline. It's like kind of a lot of extra information that you don't really need to know to understand the concepts of bioinformatics, but it is of course important as you guys are running this on clusters yourselves. And a lot of this stuff is just me being as organized as I possibly can and getting as much information as I want to out of each run. Okay, it sounds like most of your jobs are done, which is awesome. So let's review an example of the standard output. And so we want to go ahead and run this cat command, and this is going to open up the standard output for what we just run. And again, just in case some of your guys' jobs are still running, I've already ran this, so we're going to look at the standard output from what I run, and this will match exactly what your guys' are. So we can go ahead and play that. Okay, and here you will see there's the start time in seconds. So this is the date in seconds. I don't know when this second starts. So like the beginning of time? I don't know. It seems like there should be more. This is the...
It's January 1st, 1970.
Oh, thank you.
And I believe it's milliseconds. That's when Unix was rewritten.
Awesome. So the standard, I guess, is in milliseconds. So then I gave it an option in the date to do this plus %s to print out that date in seconds. And so then here is the output from my host name. So this was run on node 1 of the compute nodes. We won't get into arrays, but I ran this as part of an array. That's another cool thing that you can do with Slurm is if you're running the same job on multiple samples, you can set it up as an array and it'll run the same job on multiple samples. So as you remember, we have 6 flight samples and 6 ground control samples. So this just allowed me to run submit all 12 jobs, I should say, at the same time. So these are the outputs from up here. So I echoed fastqc version with a colon and then the command fastqc -- version. So that's the output of that command. So this was run with fastqc version 0.11.9. And even since making this set of data, which I made maybe about 5 months ago, there is a new version of fastqc already out. So this version is already old. And then I print out, so this wasn't in my script above, just because this is, again, for the array. So I printed out my sample name, just because for the array, I ran all 12 samples. And so this is just showing that flight rep 1, right? So that's the sample that we all ran. And then this here is the output when I do echo call. So this will show me the exact command that I ran. And so this is where I can check to make sure, did it properly put in my input directory? Did it properly put in, or sorry, that's my output directory. Did it properly put in my output directory? Did it properly put in my input directory? And because I had a star up here, so you see how I have flight rep 1 star fastq.gz, that star is a wildcard in command line language, and that says have all the inputs that start with flight rep 1 and end with fastq.gz. Any idea why I'd do that? You can write it in the chat or shout it out loud. Any ideas? Why I put a wildcard here? This is paired end data. Does that help? Yeah. So to make sure that I'm running both the forward and the reverse read for each sample. So I want to say this is the sample, so flight rep 1, and I have a wildcard here, so that way it will run the fastqc analysis on both my forward or my reverse read. And so that's shown here in the output data. So that's why we have 2 inputs in this call. So the first input is to that flight rep 1 R1 standing for the forward read. And then our other input is the flight rep 1 R2. And so what happens after that is it starts running. So the fastqc report starts running and the standard output looks like this. So it basically gives you a log as it's going through and doing the analysis. So it tells you that it's starting the analysis for your first input, which was our forward read. And then it basically updates you every 5% until it tells you that it's complete. And again, that was for the forward read. And then after it's done with the forward read, it'll go ahead and it'll start the reverse read. And then you'll see that same standard output. It basically gives you a log. And then at the end, it should tell you that it's complete. And then this is what that whole mess of code that I have here at the bottom ends up looking like. And so what it has is, again, the end time as the date in seconds, and then subtracting that from the start date in seconds. I get the total number of seconds it took to run this job. And then I did the calculations to give you how long it took in minutes and how long it took in hours. So this took about 17 minutes when I ran it for this job. So that's the standard output. Questions about standard output? Okay, let's take a look at what the HTML report looks like. So before we do that, in order to be able to view HTML files, we have to import a library from IPython, and so that's what this command is doing. And so now that we did that, we can run this command here called iframe, which is specific to Python, and so that's why you don't see an exclamation point. And then it's going to pull up the file, the fastqc file that was generated for our forward read. And then we could tell it how big of a window we want to see this as. So if you guys aren't seeing everything you want to see, you can kind of mess with the width and the height. So you guys go ahead and run that. And what you should see is a fastqc report. And again, if you're not seeing all of this, or if it's too small or too big, you can change the width of it. And then as you're scrolling down, you can also change the height too. Yeah, it is pretty cool. So let's take a look. I have some questions that I prompt you guys for at the end of this, but just to kind of quickly run through, and notice some caveats here. It's going to be interesting for you. So our basic statistics again, so our file name, this is our forward read, we see R1, and also we know because in our command we told it to look at the forward read. And we see our total sequences. How many sequences do we have here? That's one of the questions. And pull up in another line what these questions are. Yeah, so as we're going through this, guys, I'll ask you a question. I'm going to pull up the questions that are at the end. Actually, I don't think I ask questions until we compile these, so let's wait. So this is what it looks like just for one sample, right? So this is our forward read for our first biological flight replicate. And so we've already walked through all of this stuff. And one interesting thing you'll notice here, what's the sequence length of our raw data? Type it in the chat. Yeah, 100 to 150. Is that weird? This is raw data. We haven't trimmed it yet. So this is pretty weird. You don't want to see this in raw data. You really want to see all of your sequence lengths being the same. And the reason that this isn't the same is we actually outsourced. So this is before we had a sequencer in-house. And so when we isolated RNA for these samples, we sent it out to UC Davis for sequencing, and we wanted them to sequence it 100 base pairs sequencing length, and we wanted to have a certain depth. I think we asked for something crazy like 100 million reads. This was before my time. And basically when the data, maybe we asked for 60 million reads, and when the data came back to us, the bioinformatician that looked at it on GeneLab basically said that we don't have the sequencing depth that we paid for. And so we basically told Davis to sequence our samples deeper, so that way we get the depth that we paid for. But what they did is when they resequenced some of these samples, they added it on to another experiment. Because remember, you can pool a bunch of samples together and put them all on the sequencer at once. But it so happens that the other experiment that they combined our samples with, that needed to be sequenced at 150 base pairs. And so when they gave us back our additional sequences, the sequence length was longer than the sequence length of the original data that we got back. And so we debated, I say we, I wasn't here for this. Oh, I guess when I analyzed it, I did. Debated whether or not to keep the 150 base pair length. And normally you wouldn't. The only reason why I was okay keeping the mix of the 100 length and the 150 length is because there was the 150 reads were in every single sample. So it's not like only the flight samples had some reads with 150, because that would create bias. So because we had all of the samples had a mix primarily of 100 length with some of the reads being 150, I was okay to just keep that data and run it. So that's why you see that. That's not normal. That's not common. And usually that should raise a flag for you. But I kept it. Let me make this a little wider so we could see it all the way. So this is where it's kind of nice to see this stuff all the way. I thought I'd optimize it, but apparently not. So you could make this with, you could make this with larger or smaller, and then you'll see more or less of your information. So make this even smaller. Oh, it's because I made my screen bigger. Went in the wrong direction. Okay, we can scroll to the right, though, that's okay. So if you're not seeing your full graph of the quality scores, you guys can scroll left and right. And so taking a look, this is the forward read. So you guys are all used to seeing this from the lecture. So we're gonna see the same outputs as the lecture. We're gonna analyze them after we compile them. So this is one that I didn't show in lecture, so it's per sequence quality. So this kind of looks at the different regions on the flow cell and kind of gives you an idea of the quality that way. I don't usually use it, which is why I didn't really go over it. And then we have per sequence quality scores, which we did go over, per base sequence content, which we went over. See the per GC content. Anything you notice here? Huge peak, yeah. So there's some kind of contamination in here, probably ribosomal RNA. N-based content, so really nice. We see almost no ends. Sequence length distribution. So again, I know that it, Felix, to your point again, I guess a little smaller. We see that it goes from like there to there, which is just weird. Really, this is all 100. And then we have a bunch of them at 150. Well, I guess a smaller percentage of them. So you see the majority of the reads for all sample are at 100 sequencing length. And then we have a small percentage. Those are the additional reads that we were given that were sequenced at 150 length. Let's look at duplication levels. Any concerns here with the duplication levels? Are there duplicates here? Yeah, so we definitely see some duplications again. And again, keep in mind all the caveats associated with that. So I always take duplications from FastQC reports with a grain of salt. The other thing that I didn't go over is overrepresented sequences. So these peaks here, right, are sequences that have multiple duplicates. And so what FastQC will also print out is what the sequence is that's being overrepresented or that has multiple copies. And it'll tell you how many exact copies it has of the sequences that they're seeing like a ton of. And so one cool thing about that is it can help you kind of troubleshoot, because you can grab this and you can do a blast search on the sequence to see if it hits anything. And I think it does that internally with FastQC to kind of give you an idea of what could be the source of those duplicates. Do we have adapters in this sample? Oh, yay, all HPC jobs completed. Awesome. Yeah, we do have adapters, a little bit of them, so not too many. And where those adapters come from? Yeah, they're Illumina. This was sequenced on Illumina platform. Okay, so, is that everything? Yeah. And so we did that for the forward read. Now let's take a look at the reverse read, and we're gonna import this IPython library every time, because we notice when we practice this that sometimes importing it once it'll like stop running, and so we just want to activate it every time. So go ahead and run this next command, and then you could run the next report. And this one we're looking specifically at the reverse read. So in the example I had showed, it was only showing the reverse read. So now you saw the forward read. You also see the reverse read, and kind of as we talked about, we do see lower quality in the reverse read, again, for whatever reason, not entirely sure. So this is all the same information, except now this is for that reverse read for the same sample. And so it should be fairly similar to the, well, it's usually fairly similar to the forward, but not always. And so we do still see some contamination in the reverse reads. For the sample, a touch of N content, we should see the same exact sequence length distribution. And then again, we see those duplication levels, and it tells us what those overrepresented sequences are, so we can kind of do some extra analysis. And then you also see that we have adapters on our reverse sequence. And so we just saw the forward and the reverse data for just one sample. But remember, we have 6 flight samples and we have 6 ground control samples. And so it's really nice and convenient to be able to see what the quality looks like of all of our samples in an experiment. And so one way to do that is with this tool called multi-QC. And so we can use this program, and what it does is we tell it where to find all of those FastQC reports, and then multi-QC will compile all of the information from all samples and create a single report. And this allows us to view and assess the raw sequencing data of all samples. So let's go ahead and do that. So remember, I told you that I ran this already, so that way we don't have to go through and wait for everyone to finish analyzing FastQC for all samples. I've already done that for you. And so let's go ahead and compile this in real time. So we're going to run this command directly. It doesn't require that many compute resources, and so we should all be able to run it directly. So go ahead and start it, and you'll see what the standard output looks like. It'll be printed right out on the screen for you. Is anyone's not done yet? Anyone still waiting for their multi-QC report to finish? Okay, so you guys are all seeing what I'm seeing? Yeah, mine has the same result as you. Right, it should. So what you can kind of see from the standard output of multi-QC, this is nice. So not all tools do this, which is why I add that command to most of my scripts to tell me what the version is. But, you know, tools that are written well will, anytime you run it, it'll automatically print out the version. So multi-QC does automatically print out the version. And it also tells you if you are out of date. So again, this is another tool that has already been updated since I generated these data. It's been updated twice, actually. It's on 1.10.1. And so the version I had installed is 1.9. And so that's kind of nice because it'll tell you, hey, there's a new version of this. And so it'll search in the location we told it to search. So remember, so the parameters for this, I probably should have went over this first. The parameters for this tool, we have a -n, and for multi-QC, n tells you what you want the prefix name of your output files to be. So I had this prefix name be raw_multi-QC, so I know that this is the multi-QC of the raw quality data. And then -o, this is the output directory, so it tells where you want to store the results. So all of your outputs are being stored in your home directory in GLDS 104, 00 raw data, FastQC reports. And then remember, we made that raw multi-QC report directory within there. So all of your multi-QC data will be in that folder. And the last thing is what's known as a positional argument. So you don't give it any options. You just make sure that it's at the end of your command. And this is to point to the directory that has all of your FastQC reports that you want to compile. And so what multi-QC does is it'll search for that in that location. And so remember, we stored this as a variable. And so we should see the variable printed out. So what's stored in that variable. So this is actually the location of all those FastQC reports for all samples. And remember, we have 12 samples, right? 6 flight, 6 ground control. It'll tell you how many reports it found. It found 24 reports. Why? Forward and reverse. Yes, absolutely. Yeah. We have 6 flight samples, 6 ground control samples. Each one of those 6 samples were sequenced in forward and reverse. So yes, we have 6 forward reads and 6 reverse reads for flight samples, 6 forward and 6 flight for ground control, making a total of 24. So you always want to check and make sure that it found the number of reports you expect it to. And then it kind of walks you through again, tells you that it's compressing the data, that it's generating the HTML report, and then generating the folder containing the data that made the HTML report. And then it'll tell you when it's complete. So the output data again for this is a folder containing all of the data that's used to generate these nice graphs. So let's take a look at those graphs. And then I also have a link here. If I know I kind of went over a lot of the descriptions of the graph. We're going to see it, the multi-QC report. And so we want to click here if you want to learn more about what the multi-QC plots tell you from the FastQC data, but it's essentially the same as the FastQC report itself. So we'll take a look at this and then we'll head to lunch. So we want to go ahead, since this is an HTML file, we want to import that library. And then we want to run, we want to look at the HTML report. And so you'll notice that, remember the prefix name we gave it with the N was raw underscore multi-QC. So that's why you'll see that the output data is raw underscore multi-QC. And then it ends in .html because it's the HTML report. And as you're looking at this, I want you to kind of think about these questions, and I'll ask them as we're going. So go ahead and run that. And now you should see the same information that was in our FastQC report, except now we see it for all samples. And if you don't see all samples displayed here, you can press this arrow down. And it, is it okay if I go 5 minutes into your lunch? Is there anyone that has to hop right at noon?
I do need to leave right at noon.
Yeah, I saw your message.
Okay.
Girl, you know this stuff. Okay. So as you can see, we have some additional information here in the general statistics. So it actually tells us right up front the percent duplication and the percent GC content. And then it also tells us the number of reads in millions of sequences. And so I want you to take a look at that millions of sequences column for all of your samples. And again, you'll notice R1 is your forward read and R2 is your reverse read for each one of these samples. So you should see your 6 flight and your 6 ground control samples here. And as you're looking at those data, I want you to tell me which sample was sequenced at the greatest read depth, which sample was sequenced at the least read depth. You kind of look at this column here and put your answers in the chat for least and greatest, and it should be associated with a sample. Ground control rep 1 for most or least? Most, yes, that is right. Which sample was sequenced at the least read depth? So which sequence has the least, or which sample has the least number of reads? Flight rep 2, yeah. Yeah, flight rep 2, a lot of you guys are saying that, right. So we see that ground control rep 1 was sequenced at 85.7 million reads, and flight rep 2 was sequenced only at 65.2 million reads. So that's a difference of about 20 million reads. Is that a problem? Yeah, for sure. Yeah, Awen, you think right. Rashi, since you're the first one to answer, why do you think that's a problem? You can unmute your mic.
I feel like it won't be easy to compare against each other since there's like more reads, so it's not an good comparison. That's my best guess.
Yeah, so and it's very difficult, right? Because we're combining these samples, the libraries for these samples at the same concentration, but because there's that range, right, of insert sizes, and we know that there's a slight bias on the flow cell for smaller libraries, and so because of that, even though we're putting the same concentration of libraries into our flow cell, we don't always get the same exact number of reads. In fact, it's almost impossible to get the same exact number of reads for every single sample. So at GeneLab, we do quality control on our libraries. We sequence it at a much smaller depth to kind of look and see if it's called balancing your library. So you really want to make sure that you're getting as close as possible to the same number of reads per sample. However, because it's literally impossible to do that, there is always going to be some differences, and sometimes you'll see greater differences than others. Hopefully you won't see too great of difference, but you're absolutely right. If we have a sample that we sequenced more reads with than another sample, just by matter of we have more information from one sample than another, so we might see a higher expression of a gene in one sample that we sequence greater than the other, which is not true. We just have more reads, right? And so this is another thing when we do our normalization. In addition to accounting for the transcript length, we also have to account and normalize for the number of reads we have per sample. How do we exclude one of the Oh yeah, so no, good question though. That is one way you could do that, but the goal is to not reduce your number of biological replicates. So yeah, we actually account for that with math. And so we do some normalization to account for the total number of reads for each sample. But that wasn't a bad guess. Okay. Moving on to sequence counts. So this is another way to look at the overall sequence counts. And this is showing us duplication. And so this is pretty cool that we can actually see the raw number of reads. So you see number of reads here. Or if we want to see percent of total, since we know, of course, the number of reads are going to be different for each sample, we could look at the overall percentages, and then we can see what percent of the reads are unique and what percent are duplicates. Again, this is the caveat that comes with how FastQC determines duplicate reads. And then looking at the sequencing data here. So this, now we have all of our samples together. And so it kind of graphs them in a line, and you can hover over. As you noticed, when I hover over, it'll tell you what sample you're looking at. And what do you notice about all the samples down here? Are they forward or reverse? Yeah, they're all reverse. What about all the samples up here? All the ones up here are forward. Yeah. So what does that tell you about the quality of the forward reads compared with the quality of the reverse reads? Yeah, forward better quality. Diksha, you are ahead of me. So again, we're seeing consistently for all of our samples, we notice that the forward read has slightly better quality than the reverse read. And but overall, how do you think the quality is? Good quality, bad quality? Yeah, pretty good. It's actually pretty good quality. Okay, we're going to cover 2 more questions and then head to lunch. So this again is just, the other way of looking at the quality, this time, of course, for the entire sequence as opposed to the base position. This is looking at that per base content. It just tells you the breakdown. So this is how it compiles. So do you remember those graphs that had the red, blue, green, and I can't remember the other color, lines for your A, T, C's, and G's? So this is kind of how it compiles it all together. And it'll tell you the breakdown. So as I'm scrolling, you'll notice up here, you'll see the sample and it'll tell you the breakdown. And then this says that it fails. So there's some arbitrary cutoffs that multi-QC has for compiling FastQC data. I don't really pay too much attention to that personally. And this really fails because you know how at the beginning there's that wobble? And that's just for a caveat of RNA-seq data. Multi-QC doesn't know that this is RNA-seq data. And so those cutoffs, I think, are just for sequence data in general. So in general, you don't really want to see that wobble at the beginning. And so they have these random fail and warning cutoffs that they have. And if you want to learn more about that, that's why I provided the multi-QC link here. So if you click there, you can read all about the description of the multi-QC plots derived from FastQC data. So So you can read more about what those cutoffs are and what they mean. Again, we see the graphs for the per GC content. So we're noticing that all of them have in their forward and reverse reads this kind of spike over here, showing some contamination. And then let's just take a look at again, with our read length. So we see for all of the reads, as I mentioned, every single read has a mix of both 100 base pair length and 150, which is why I was okay moving forward with them. Again, we see duplication in all samples, and the duplication looks pretty consistent in all samples, which again makes me think that a lot of that's ribosomal contamination. And then adapter content. Do we see adapters in all samples? Yeah, for sure. Although we do see it, it still is a very small percent, 3% of the sequences really, in most samples or less. It is nice to get rid of that. And so I guess my next question, and then it kind of compiles all the information from the different statistics that it gives you. And again, these green, red, and yellow are based on the cutoffs for multi-QC, which you guys can look at by looking at the link. So it kind of compiles all the information together, which is nice and convenient. And then we answered all these questions all the way, except for this last one. Should we trim or filter the data before aligning to the reference genome? Yeah, for sure. So we will trim and filter our data when we get back. So go ahead and enjoy your lunch and be back here by 1 p.m.